All right, you guys should have your notebooks out and you should be ready to take notes. I am unfortunately sick today, so that's why I'm doing these notes for you guys. Your title here is DNA. Most of you should have that title down from yesterday with the exception of honors. Honors, we didn't get to the notes yesterday. I will catch you up when I get back. So you should have written down already that DNA stands for deoxyribonucleic acid. If you don't, you need to write that down right now. We talked about how there's different parts of words in here. We said the goal of DNA is to have the instructions on how to make proteins. So we have the word ribo here, which stands for ribosome, nucleic, which stands for nucleus, and then deoxy. So I showed some of you that yesterday that it's missing an oxygen. So normally the sugar that's in DNA has oxygens on all 5 of its sides, but it's missing one oxygen. That's why we call it deoxy, because it's missing one oxygen. And it's found in the nucleus of all cells. So you probably remember the animal cells and the plant cells we talked about, right? Remember the polka dots? Polka dots are the ribosomes. Those make the proteins. We talked about the endoplasmic reticulum transporting proteins and the Golgi body transporting proteins. So the nucleus holds the DNA. And if you remember, a lot of times we talked about this DNA as an X shape because that's the shape it's in when it's wound up in a chromosome. Okay. So our DNA, remember, looks like a double helix. All right, so we have that double helix, and it'll look like an X when it's wound up into chromosomes, but a lot of times it just looks like a big ball of spaghetti because it's not wound up in chromosomes, and we will get to that. So, reminder, DNA contains the instructions for making protein. You absolutely 100% need to know that, okay? So make sure you write this down. Just like how a chocolate chip cookie recipe has the instructions for making chocolate chip cookies, DNA has the instructions for making proteins. Now, if you know anything about recipes, you know we can make different types of cookies. Not all cookies are the same. And just like that, we can also make different types of protein. Yesterday, we talked about how there's different types of proteins. There are proteins that help form structures in your body. There are proteins that help transport things. There's proteins that help make up blood and your muscles and hormones and enzymes. So lots of different types of proteins. Here are some of the examples. You do not need to write down anything from this slide, okay? But here are some of the examples. We have contractile proteins. They can contract. This is what makes up your muscles. We have insulin or other hormones. Those are made of proteins. Antibodies. These things help fight off viruses. I hope they are working in my body right now because I am not feeling too hot. Hemoglobin. This is what makes up your blood and helps you transport oxygen. Enzymes. These speed up chemical reactions. Some of them store stuff. Okay, this is storing iron. It could be structural, like spider silk is made out of a protein. And it can be a receptor. A receptor is kind of like it intercepts signals from other cells, kind of like a cell phone tower. It receives messages. So lots of different types of proteins. Now, remember we talked about a monomer. A monomer is a building block. Remember when we talked about macromolecules? We talked about lipids, we talked about carbs, we talked about proteins, and then the last one was nucleic acids. Okay, so a refresher here. The monomer of lipids, anyone remember? Starts with an F. I might as well just type this. You don't need to write the bottom part here. You just need to write the top. So the monomer of lipids was fatty acids. Okay. We also talked about carbohydrates. Anyone remember the building blocks of carbohydrates? We said they were sugars or monosaccharides or glucose. Either one works. When we talked about the monomers of proteins, anyone remember what proteins are made up of? They're made up of amino acids. Again, you don't need to write this bottom part here. This is review. What I need you to write is the top part. So the building blocks of DNA are called nucleotides, which makes sense because it's in the nucleus. Well, let me show you what a nucleotide looks like. If you look at this picture, you can see that there's lots of different shapes that are making up this DNA molecule. So this is a nucleotide, one nucleotide here. And we have lots of different nucleotides making up this molecule, right? One on each side. So what the heck is a nucleotide then? Well, as you guys saw in that worksheet you just did, a nucleotide is made up of 3 different parts, okay? So each nucleotide is made up of 3 parts. The first part is a sugar, and this sugar is called deoxyribose. I'll let you write that. So it's a pentagon shape. Anything that ends in ose, like glucose or fructose, is a sugar. Now remember we said deoxy, that means it's missing an oxygen. So here we're supposed to have an oxygen. Notice that each one of these vertices, right, there's an oxygen, has an oxygen. Here is the oxygen, here's the oxygen, here's the oxygen, but this last side or vertex doesn't have an oxygen, so that's why they call it deoxyribose. And it's a pentagon. That's the shape the sugar is. So it's made up of a sugar. It's made of a phosphate group, okay? Phosphate has phosphorus and oxygen. Don't worry about memorizing this specific thing. You just need to know that it's a phosphate group. And as you can see down here, the phosphate group is represented by a circle. Now this isn't exactly what a nucleotide looks like. It's a lot more complicated than these little shapes, but we like to simplify things. The last thing that makes up a nucleotide is a base. Okay, notice this green rectangle is the base. The base can be one of 4 letters, A, T, C, or G. Remember how we talked about there being a code? Well, those letters are the code. And if we rearrange those letters in different orders, they tell the cell to make different proteins. So just like a computer code has zeros and ones in specific orders, or just like Morse code has dots and dashes in different orders. These letters, these 4 letters, are the code. So what I want you to do is in your notes, we're going to draw a picture of a nucleotide. Okay, so you can draw it below here, or if you have room on the side. So I want you to draw a pentagon. Now, if you're like, I don't know how to draw a pentagon, draw a house. That's all you got to do. Draw a house. Okay, so everyone should have a house. This is the sugar. So you want to label that sugar. And it's pretty impossible for me to write on here, but I will try my best. So sugar. All right. Now, what I want you to do is draw a circle. This is the phosphate. Okay, so label that. I'm just going to put a P. You can label it phosphate. And then we're going to make a rectangle. And we're going to label that our base. Remember, our base can be one of 4 letters. Okay, pretend that says base because I really can't write. A, T, C, or G. So now we have a little drawing of our nucleotide. Now let's look at what this nucleotide actually looks like. I'm going to Google a picture because I want you to see this. I'm going to Google nucleotide molecular structure. Because a lot of times in science, what happens is we oversimplify things, which is great for learning, but sometimes it's important to know what these actually look like. So here you see a picture of an actual nucleotide. This is what a real nucleotide looks like. Okay, so even though we draw the phosphate as a circle, it's not really a circle. And the sugar is a pentagon. The base isn't really a rectangle, but we simplify things, okay? So many nucleotides then join together to create a double helix. This is what we talked about yesterday with Watson and Crick, okay? So the nucleotides here, I'll circle them. 1, 2, right? Notice there's one on each side. And these nucleotides join together and they make that spiral double helix. We talked yesterday about Linus Pauling thinking it was a triple helix. Okay, honors, don't worry, I will catch you up. We didn't talk about that yesterday. But some people thought it was a triple helix, but it turns out it was just a double helix. Okay. So what then makes up this helix? Okay, we know they're nucleotides, right? Let's look what's in the middle. In the middle, we have our bases, our letters A, T, C, G. On the outside, we have our pentagons. Remember, those are our sugars, and the phosphates. So what we see here is that the sides of the helix, the sides are made of sugar and phosphate, and the center is made of bases. Okay. You don't need to write this down. If you would like to, you can, but I would expect that from looking at a diagram, you should be able to tell that the sides are made up of sugar and phosphate. The center is made up of bases, okay? So how do these bases match up? They match up in certain ways because if you look back at this diagram, there's always two bases in the middle, and they can only match up in certain ways. So the bases always pair up in the same way. So here you can see, again, these are simplified, but adenine always pairs with thymine. Okay, these are bases, the letters A and T, right? So A always pairs with T. Again, if you don't want to write the full name, just write A bonds with T. And then cytosine, C, bonds with G. They always bond this way, okay? And again, the order that they go in down this molecule, the order the letters go in, is the code that's going to code for the different proteins. So A always bonds with T, C with G. There's no other way it can happen. Now, occasionally it makes mistakes. There are mistakes. Sometimes we have a mutation, okay? We'll talk about that. And a mutation is just when we get a different letter here. Sometimes it's bad. A lot of times it's not bad because what I haven't told you is that a lot of our DNA doesn't code for proteins. A lot of our DNA is just junk, okay? It would be like if think about reading the phone book, okay? Um, Usually you're only looking for one or two phone numbers in there. The rest of the phone numbers you don't care about. They're kind of junk, okay? So some of our DNA is junk. It doesn't code for proteins. So if we get a change in a letter and it doesn't code for a protein, well, it doesn't matter. So you'll notice in the beginning here, or in the middle, they're going to be connected by what we call hydrogen bonds. So in the middle, we have these bases, okay, and they're connected by hydrogen bonds. So I'm going to have you write that in 1 second here. So apparently I do not have a slide for that. So what I'm going to do is I'm just going to make one. So the bases are connected by hydrogen bonds. So I want you to write that down. In the middle, they're connected by these hydrogen bonds. I'm going to grab a picture. I will come right back to this. And apparently I can't copy and paste there. So just know that they're connected by hydrogen bonds. And we can see that if we go, and I'll show you these pictures in a second. Let's see if I can copy them again. There we go. So this is adenine and thymine, okay? These little dashes are showing how they're connected. It's two bases and they're connected by bonds. You guys know about bonds. You've probably heard of covalent or ionic bonds. And you've probably talked about bonds or electrons maybe in 8th grade. Well, these are called hydrogen bonds. The reason called hydrogen? Well, it's hydrogen that's connecting to another atom, okay? So we have A and T, and if you notice, there's different number of hydrogen bonds. So how many bonds are there between A and T? 2, because we see 2 dashed lines. How many bonds are there between C and G? If we look at that, how many dashed lines do you see? We see 3. So that's why A and T can pair with each other and C and G can only pair with each other. If I tried to take C, it wants to form 3 bonds. It wouldn't be able to pair with like A, okay? Because they want different numbers of bonds. All right, and then last but not least, this is the last thing we're going to write. So the DNA strands, we call them anti-parallel. Let's think for a second, what does parallel mean? I'll put that back in a second. What does parallel mean? Parallel means they never run into each other, right? Like train tracks, okay? Remember from math, perpendicular is like a T, parallel is like this. Now, anti-parallel means one strand, goes one direction, the other strand goes in the other direction. So look at this. You guys see how one strand, the phosphate, is pointing up here? On this strand, the phosphates are pointing down. Or if we look at the house, right? The pointy part of the house is pointing up here. It's pointing down here. So our strands run in opposite directions, right? One strand is pointing one way and one is pointing the other way. Sometimes you'll see them labeled as 5 and 3 ends. We call these 5' and 3'. Again, not super necessary that you know this, but if you notice the 5 part of one end matches with the 3, right? So this side it goes 5 to 3, this side 3 to 5. So they're going in opposite directions. That's all you need to know. And this is called anti-parallel. So I'll give you a second to write that down. So honors, if you have any time, what I'm going to have you guys do is I'm going to have you color code your DNA molecules. Regular, you probably won't have enough time. In the event that you do, here are the colors you're going to be coloring them. So you'll need these colors. Use markers, crayons, colored pencils, whatever you want. Color them. We're going to cut them out tomorrow. If you want to get a head start on cutting them out, that's fine. I can leave like some plastic bags, but I'm guessing you won't get that far, so color them, hang on to them. If you still need to work on coloring them tomorrow, that's totally fine. I should be here tomorrow, and that's pretty much it. So have a good rest of your day. Be nice to the sub, and I will see you all tomorrow.